In this presentation, we will describe the workflow that was adopted and the results that were obtained in two studies where we investigated brain dynamics in brain tumor patients, before and after tumor resection using the virtual brain. More information can be found in these papers. As you can see in this short video, it is evident that there are quite some important neural elements close to the tumor that should be spared during surgery in order to preserve important functions. When planning to resect a tumor, a neurosurgeon will typically rely on this kind of information. Although this information is very useful, it is insufficient to predict the results of neurosurgery at the individual patient level. This is because the brain is a very complex system that is slightly different for each individual, and even more so as a result for disease processes such as infiltration with a brain tumor. Therefore, computational modeling is a very promising approach, particularly in the context of brain tumor surgery, as it would allow us to experiment with different neurosurgical approaches by inducing different types of lesions to the brain structure, after which we can simulate resulting brain dynamics for each scenario and choose the optimal one for the actual surgical procedure. In other words, we could try to build a neurosurgery simulator. For our studies, two types of brain tumors were collected. Meningioma tumors grow from the meninges. These generally don't infiltrate the brain, but they can exert pressure on the brain as they grow larger. Glioma tumors, on the other hand, grow from the glia cells within the brain. In addition, a group of healthy controls was recruited consisting of the primary caregivers of the patients. On the day before each patient's tumor resection, the first test moment took place. At this moment, different MRI images were acquired, cognitive performance was assessed, and the participants were asked to complete several self-reporting questionnaires. Several months after the patient's tumor resection, when they attained stable neurological functioning, all measures were repeated. Of note, all data are publicly available, both in raw format as well as processed structural and functional connectivity matrices ready to use in the virtual brain. For our simulations, we have used the reduced Wong-Wang model. This is a dynamical mean field model that summarizes realistic dynamics of detailed spiking and conductance-based synaptic large-scale network. Two different model parameters were individually optimized. Global coupling G, global parameter, describing dynamics between brain regions. Feedback inhibition control parameter JI, describing the amount of inhibition within each brain region. This is a local model parameter. This allowed us to study possible differences in regions close to the tumor and healthy regions. In the first study, we then explored whether we can use the same model of brain functioning for healthy subjects and brain tumor patients. In particular, we investigated optimal model parameters in the brain tumor patients before tumor surgery compared to healthy controls. To this end, we conducted a parameter space exploration. Specifically, we simulated brain dynamics with a range of parameter values for the global coupling and compared the resulting simulated functional connectivity matrices to the empirical functional connectivity to identify the parameter value that maximized the correspondence between both. Of note, the regional feedback inhibition control parameters were tuned automatically in each iteration of the parameter space exploration to clamp the average firing rate at 3 Hz for each excitatory mass model. As a final step, we then evaluated whether these optimal model parameters differed across groups and whether associations with other relevant variables were present. As a final step, we then evaluated whether these optimal model parameters differed across groups and whether associations with other relevant variables were present. Two elements contribute to the construction of personalized virtual brains: individual SC matrix and fitting of model parameters by using the individual SC matrix. Using individual structural connectomes did not yield better predictions of individual FC patterns compared to using the control average SC matrix. This is, however, a limitation of all current computational modeling studies. A recent study by Zimmerman and colleagues focused on this exact issue and reported that the subject specificity of SC-FC is limited due to the relatively small variability between subjects in SC compared to the larger variability in FC. This limited variability in individual SC matrices could be due to the quality of current individual SC matrices. To overcome this limitation, data of even better quality or finer and more elaborate parcellations For example, based on multimodal data such as Glasser et al. 2016, could be used to investigate whether individual traits can be captured through tractography that have relevance for the prediction of individual FC. Secondly, our results have shown that model parameters differ significantly between healthy controls, left box plot, and brain tumor patients, middle and right box plot. In particular, the amount of inhibition was higher in regions distant from a tumor, relative to healthy brains, whereas it was much lower in regions closer to the tumor. No significant group differences were obtained for the global coupling value. Third, model parameters were found to correlate with structural network topology and cognition. Specifically, the global scaling parameter was related to the global efficiency of the structural network, a relationship that has been found in stroke patients as well. In addition, local feedback inhibition across non-tumor regions in patients was also related to the structural network global efficiency. Finally, we also found some associations between regional feedback inhibition across tumor regions and patients' cognitive performance. But as you can see in the bivariate scatter plots, the associations were not nicely linear, so more data would be required to confirm these relations. In the second study, we then repeated the same analysis procedure on the data acquired after surgery to investigate the stability of model parameters over time and after surgical intervention. In contrast to our expectations, results showed that model parameters were relatively stable over time in patients who underwent tumor resection, relative to baseline variability levels observed in healthy controls. In addition, the associations between model parameters and structural network topology and cognitive performance were relatively stable over time. Since the models appeared to be patient-specific and relatively stable over time, this allowed us to construct personalized virtual brain models based on the patient's pre-surgical data and to evaluate whether post-operative brain dynamics could be predicted by virtual neurosurgery. As proof of concept, we performed these proof of concept analyses in 7 glioma patients for which we had both pre- and post-operative data available. In particular, we performed virtual neurosurgery mimicking the actual neurosurgical procedure, after which we simulated brain dynamics using the patient's individually optimized model parameters. As a final step, we then evaluated the accuracy of the predicted brain dynamics after virtual surgery by comparing them to post-operative empirical functional connectivity. This figure shows the results of our virtual neurosurgery analysis in green, and those of the preoperative model explorations in blue. Now looking only at the green data points, our analyses show that promising results for 3 out of the 7 patients. Importantly, model performance during preoperative parameter exploration appeared to be a key indicator for the potential of this technique for virtual neurosurgery. In particular, prediction of post-surgical brain dynamics seems to be feasible only in patients for which the model also substantially improves prediction accuracy beyond the structural connectome during parameter exploration. Specifically, out of the 4 patients for which this was the case, prediction accuracy of post-operative brain dynamics is relatively successful in 3 of them. In the other 3 patients for which computational modeling only resulted in marginal gains in prediction accuracy relative to the structural connectome, so when the difference between light and dark blue is relatively small, results show worse prediction accuracy. Nevertheless, for those patients, the virtually lesioned structural connectome serves as a good approximation of their post-operative functional connectivity, suggesting that their brain dynamics are more determined by the underlying structure in the months following the tumor resection. To allow reproduction of these results, we have provided an IPython notebook with some example code. As a first step, all required packages are loaded. Subsequently, individual structural and functional connectivity data are loaded. The input data has to follow the directory structure as is described in the IPython notebook. All data that is used for this study can be found at this GitHub URL. Next, the simulator is configured. Here you only have to adjust the repetition time of the bolt signal you want to simulate, corresponding to the TR of the RS fMRI sequence, monitors.temporal average and monitors.bold, and the simulation length of the simulated brain dynamics in milliseconds. Finally, you can run the simulation with a user-defined range of values for the global coupling strength, or GC range. The plot shows the correlation between simulated and empirical FC for different values of the global coupling. Thank you for watching this presentation. We hope that you have found it educational and useful.